Good afternoon to all. In today's lecture, we have discussed about image formation and the structure of human eye. First, we have to study about elements of visual perception. In particular, our interest in the mechanics and the parameters related to the how images are formed and the perceived by humans. We are interested in learning the physical limitation of human vision in terms of factors that are also used in our work with the digital images. Then we have to study about the structure of human eye. The eye is nearly a sphere with an average diameter of approximately 20 mm. There are three membranes encloses the eye that are cornea and sclera. Sclera is the outer cover, choroid and retina. We have discussed one by one. First one is a cornea. The cornea is a tough, transparent tissue that covers the anterior surface of the eye. Continuous with the cornea, the sclera is an opaque membrane that encloses the remainder of the optic globe. Next one is the choroid. The choroid, it contains a network of a blood vessel that serve as the major source of nutrition to the eyes. It helps to reduce the extraneous light entering in the eye. It has two parts: iris diaphragm and ciliary tube. This membrane contain or this choroid coat is heavily pigmented and has helped to reduce the amount of extraneous light entering the eye and the backscatter within the optical globe. This membrane contains the network of blood vessels that serve as the major source of nutrition to the eye. The choroid lies directly below the sclera. At its exterior extreme, the choroid is divided into the two parts, two membrane, that is ciliary tube and iris diaphragm. The latter the contract and or expand to control the amount of light that enters the eyes. The front of there is contains the visible pigment of eyes, whereas the black contains the black pigment. The central opening of iris or the pupil varies in the diameter from approximately 2 to 8 mm. The front of iris contains the visible pigment of the eyes, whereas the black contains black pigment. The lens is made up of concentric layer of fibrous cells and is suspended by fibers that attach to the ciliary body. It contains near about 60 to 70% of water and about 6% fat and more protein than any other tissue in the eye. Then next one is the retina. The retina is the innermost membrane of the eye which lines the inside of the wall's entire posterior portion. When the eye is properly focused, the light from an object outside the eye is imaged on the retina. The pattern vision is afforded by the distribution of a discrete light receptors over the surface of the retina. There are two classes of receptors, that are cones and rods. The cones in each eye is number between 6 to 7 million. They are located primarily in the central portion of the retina, called as the fovea, and are highly sensitive in a color. These are highly sensitive to color. Human can resolve fine details with these cones because each one is connected to its own nerve end. Cone vision is called photopic or bright light vision. Muscle controlling the eye rotate the eyeball until the image of an object of interest falls on the fovea. Cone vision is also called phototopic or bright light vision. Then next one is the major class of the receptor are rod. Rod are much in number from 75 to 150 millions and are distributed over the entire retinal surface. The large area of distribution and the fact that several rods are connected to a single nerve give a general overall picture of the field of view. They are not involved in the color vision and are sensitive to the low level of illumination. Rod vision is called scotopic or dim light vision. The absence of Reciprocators is called blind spot. The figure shows the distribution of rod and cones in the retina. Then the absence of a receptor in this area result in the so-called blind spot. The figure shows the distribution of the rods and cones in the retina. We have to seen earlier. Then next topic is the image formation in the eye. The principal difference between the lens of an eye and an ordinary optical lens is that the former is flexible. The shape of the lens is controlled by the tension in the. Then next one is the image formation in the eye. The principal difference between the lens of an eye and the ordinary optical lens is that the former is flexible. The shape of the lens is controlled by the tension and in the fibers of a ciliary tube. The focus on distant objects, the controlling muscles cause the lenses to be relatively flattened. Similarly, these muscles allow the lens to become thicker in order to focus on objects near the eyes. The distance between the center of the lens and the retina are called the focal length, and it varies nearly approximately from 17 mm is to about 14 mm as the refractive power of the lenses increases from its minimum to its maximum. When the eye focuses on an object farther away, the lens exhibits its lowest refractive power. When the eye focuses on a nearby object, the lens is more strongly refractive. For example, the observer is looking at a tree 15-meter height at a distance of 100 meter. If h is the height in mm of that object in the retinal image, the geometry figure as shown in figure yields 15 divided by 100 is equal to h upon 17 or h is equal to 2.55 mm. Perception then takes place by the relative excitation of a light receptor, which transform the radiant energy into electrical impulses that ultimately are decoded by the brain. Then next one is the brightness, adaptation and discrimination. The range of light intensity level to which the human visual system can adapt is of the order of 10 raised to 10 from the scotopic threshold to the glare limit. The human eye is wide dynamic range by changing the eye's overall sensitivity, and this is called brightness adaptation. Then the figure shows the range of subjective brightness sensation showing a particular adaptation level. The HVS means human visual system mechanism adapt to different lighting conditions. The sensitivity level for a given lighting condition is called as brightness adaptation level. As the lighting condition changes, our visual sensory mechanism will adapt by changing its sensitivity. The human eye cannot respond to the entire range of intensity levels at a given level of sensitivity. For example, if we stand in a brightly lit area, we cannot discern details in a dark area since it will appear totally dark. Next one is our photoreceptors cannot respond to the low level of intensity because the level of sensitivity has been adapted to the bright light. Then the figure shows the basic experimental setup used to characterize brightness discrimination. At the given adaptation level, the eye cannot discriminate between the too many intensity levels. But by varying the adaptation level, the eye is capable of discriminating a much broader range of intensity level. Then the next figure shows the typical Weber ratio as a function of intensity. Here we plot the graph of log I versus log del I C upon I. We discuss one by one what is del I, what is I. A plot of log del I upon I as a function of log I has the general shape as shown in a above figure. This shows brightness discrimination is poor at low level of illumination and improves significantly as background illumination increases. Del I is not bright. The subject says no, indicating no perceivable changes. As del I gets stronger, the subject may give a positive response of S, indicating the perceived changes. Finally, when del I is strong enough, the subject will give a response of S all the time. The quantity del IC upon I, that is del IC, is the increasement of illumination discriminable 50% of the time with the background illumination. I is called Weber ratio. The small value of del IC upon I means that the small percentage change in the intensity is discriminable. This represent good brightness discrimination. Conversely, the large value of del IC upon I means that the large percentage change in intensity is required. This represents poor brightness discrimination. If the background illumination is held constant and the intensity of the other source instead of flashing is now allowed to vary incrementally from never being perceived to always being perceived, the typical observer can discern a total of 1 of 2 dozen different intensity changes. The net consequence is that the eye is capable of much broader range of overall intensity discrimination. Two phenomena clearly demonstrate that perceived brightness is not a simple function of intensity. The first is based on the fact that the visual system tends to undershoot or overshoot around the boundary of region of a different intensities. Although the intensity of strips is constant, we actually perceive the brightness pattern that is strongly scalloped near the boundary. These seemingly scalloped bands are called as match bands after Ernest Mach, who first described the phenomena in 1865. The second phenomenon called simultaneous contrast. We discuss one by one. Then next one is the match band effect. Match band effect. The figure shows the match band effect. The image shown consists of two regions, one towards the left and the one towards the right, which are of uniform intensity. At the middle there is a strip on which the intensity changes uniformly from the intensity level on the left side to the intensity level on the right side. If we observe carefully, we notice that the dark band immediately to the right and the middle strip and the light band immediately to the left of the middle strip. Actually, the dark or the light band has the same intensity level as the right or left part of the image, but still we perceive it darker than that. This is called as a match band, or this is called as a match band illusion. This is called as a match band illusion. The figure shows match band effect. Then next one is a simultaneous contrast. Simultaneously contrast means what? The perceived brightness of a region does not depend on the intensity of the region, but on the context, means background or surroundings on which it is seen. All the center square have the exactly same intensity. The four squares are showing in the figure have the same intensity. However, they appear to the eye to become a darker as the background gets lighter. Then next one is the figure shows the electromagnetic spectrum, in which it shows the rays like ultraviolet, infrared, visible spectrum, gamma rays, X-ray, microwaves, radios, waves, etc. We study deeply. The light as we see it illuminating the object is a very small portion of the electromagnetic spectrum. This is the visible color spectrum which can be sensed by the human eye. Its wavelength span between 0.43 mm for the violet to 0.79 mm for red color. The wavelengths outside this region correspond to radiations which cannot be sensed by human eye. For example, the ultraviolet rays, the X-rays and the gamma rays have progressively shorter wavelengths, and on the other hand, infrared, microwaves and radio waves have progressively larger wavelength. Then next one is image sensing and acquisition. What is meant by image sensing and image acquisition? Image created based on two factors, that is illumination source, and next one is a reception or absorption of energy from the source by the elements of the seen being image. These type of images are generated by the combination of an illumination source and their reflections or absorption of energy from that source by the element of seen being image. Examples of the illumination source are radar, infrared, X-ray, ultrasound or computer generated illumination pattern. Examples of seen elements or objects are buried rock formation or a human brain or a sun. Depending on the nature of the source, illumination energy is reflected from or transmitted through the object. An example in the first category is the light reflected from the planar surface. An example in the second category is when the X-ray pass through the patient body for the purpose of generating a diagnostic X-ray film. In some applications, the reflected or a transmitter energy is focused on a photo converter. For example, phosphorus screen, which converts the energy into visible light. Electron microscopy and the some applications of gamma imaging used in this approach. The idea is simple. Incoming energy is transformed into the voltage by the combination of input electrical power and the sensor material that is responsible to the particular type of energy being detected, and the output voltage waveform is response of a sensor, and the digital quantity is obtained from each sensor by digitizing its response. In this section, we look at the principal modalities for image sensing and generation. Then next is imaging sensors. There are three types of imaging sensors, that are single imaging sensor, line sensor or arrow sensor. Image acquisition using the first we have discussed single sensor. This is the figure of component of single imaging sensor. Then the most common sensor of this type is the photodiode, which is constructed of silicon. We know what is a diode, what are their characteristics, and diode is made up of silicon materials and whose output voltage waveform is proportional to light. The use of filter in front of the sensor improve the selectivity. For example, a green pass filter in front of the light sensor favors the light in the green band of the color spectrum. In order to generate a 2D image using single sensor, there have to be a relative displacement in both the x and y direction between the sensor, the area to be imaged. The next one is the image acquisition using the sensor strips, line sensor. In line arrangement of a sensor in the form of sensor strip is used much more frequently than the single sensor. The strip provides imaging element in one direction. Motion perpendicular to the strip provides imaging in the other direction. This is the type of arrangement used in most flat bed scanner. Sensing devices with 4000 or more inline sensor are possible. Inline sensor are used in airborne imaging application. The light sensor are used in airborne imaging applications. Then next one is image acquisition using the sensor array. The figure shows the example of image acquisition process. A simple image formation model. A simple image, in that it includes a simple image formation model. Image denoted by two-dimensional functions, that is f of xy. We had earlier discussed what is meant by digital image, what are the coordinates of digital image. There are two coordinates and it's a function of or मतलब intensity of the दिस कोऑर्डिनेट्स. हमने देखा था कि x और y एक्सिस का कॉम्बिनेशन मिला कि इमेज को हम डिनोट करते हैं इन द फॉर्म ऑफ कोऑर्डिनेट्स f ऑफ xy. द वैल्यू ऑफ एम्पलीट्यूड ऑफ f at xy positive scalar quantity. It is a positive scalar quantity. Image generated by the physical process intensity value are proportional to the energy radiated by the physical source. It is a proportional to the energy radiated by the physical source. Is less than or equal to 0 or up to the infinite or greater than or less than infinite. F of xy may be characterized by two components, that are, first one is the amount of source illumination incident on the scene, and next one scene illumination ixy, it is denoted by ixy, and the second one is the amount of illumination reflected by the object of the scene, that is a reflectance, that is r of xy. I is a function of xy and r is a function of xy. F of xy is equal to I of xy into r of xy. F of xy is a function of both, that is illumination and reflectance, where that is the values of I of xy is lies between 0 to infinity, and also the values of r of xy is also lies between the 0 to 1. The total absorption and the total reflectance, it is called as a total absorption and total reflectance, and its value is lies between the 0 to infinite. Typical values of reflectance R of xy. Intensity 1, L minimum is less than equal to 1, less than equal to L maximum, means it lies between L minimum is less than 1 and L maximum is greater than 1. In practice, L minimum is equal to I minimum and L maximum is equal to I maximum. R maximum means what? It is a combination of both I and R. A typical limit for indoor values in the absence of addition illumination. L minimum is equal to 10 and the L maximum is nearly equal to 1000. L minimum and L maximum is called as the grayscale or the intensity scale. It's called as a grayscale or intensity scale. In common practice, shift to 0 to L minus 1, where L is equal to 0 is considered black and L is equal to L minus 1 is considered white. Means L is equal to 0 for black and L is equal to 1, L is equal to L minus 1 is considered for the white. Thank you so much, guys.